Hey, did you study for the midterm yet?
The one on quantitative genomics? I honestly don't even know where to start.
Yeah, it's a lot. I think the major concepts are the Hardy-Weinberg equilibrium, disrupting factors like non-random mating and gene flow, and the pedigrees. Don't forget about the pedigrees.
Right, so counting pathways, calculating coefficients. Got it. Would you be down for a study session after class?
Yes, for sure.
Let's start with the Hardy-Weinberg equilibrium. This equilibrium essentially assumes that allele frequencies remain relatively constant over time when passing down from generations. From this, we get the equation 1 equals p plus q squared, which we can expand. Knowledge check. What do each of these squared variables mean?
P squared and q squared refer to the proportion of individuals with a homozygous genotype.
How about the 2pq notation?
2pq refers to the proportion of individuals with a heterozygous genotype, and that'll be important to remember when we get back to calculating the carrier risk.
Exactly. The equation is pretty straightforward when you have a population that adheres to the assumptions. So this generally works if the population is large, there's random mating, and there aren't any disturbing forces. But we know humans don't exactly follow these rules, so how can we handle those special case scenarios?
Like you mentioned, one of the main assumptions is a large population and no migration, which accounts for genetic diversity and prevents genetic drift. Genetic drift is a phenomenon that refers to random changes in the frequencies of alleles across generations.
The opposing and balancing force to drift is flow. Gene flow is the spread of allele frequencies from differing populations.
A main example is the founder effect, and that happens when a small group of a larger population gets isolated, forming a new population. Because they carry a fraction of the original diversity, there's a reduction in genetic variability. For example, let's say that we have a thriving population of 20 fish, but then 5 of them get separated by a shipwreck. The 5 fish are still going to reproduce, but now because there are less of them, there's less genetic diversity. So, some traits that were rare back in the big population may become very common now, and some traits that were common before might not exist here at all.
Wait, so how does this play a role in humans?
Well, they contribute to founder variants. These are variants that occur at a higher frequency in isolated populations because they stem from a common ancestor who acted as the initial carrier. Kind of like that case we studied in class on the founder mutation in the NEFH gene that was associated with Japanese nationwide patients with CMT and SMA.
And what are their inheritance patterns?
The mode of inheritance in all 4 families was autosomal dominant. I have the family pedigrees here, but before we get into that, you do remember how to read a pedigree, right?
I think so. How about a super quick quiz?
Alright, let's go through some examples. What does this dotted line represent?
That represents adoption, so individuals who are part of the family but don't share genetic material.
Yeah, exactly. What about this diamond over here?
The diamond represents unspecified sex of an individual. You might see this in cases where an individual doesn't identify as male or female. You will also see this when performing risk calculations for an unborn child with unspecified sex.
Right, and it's important to remember that sex is primarily used to assess non-autosomal traits. Let's take X-linked recessive traits as an example. Let's say we have an affected mother and a non-carrier father. A female offspring will be a carrier. In comparison, a male offspring will be affected. In this case, what does the double line indicate?
It represents consanguinity. Keep note of this one for later. Now that we got that refresher out of the way, let's get back to the founder variant case study you were talking about.
Researchers found one specific mutation on the NEFH gene in 4 different families, all in one southern Japanese town. Each family was either affected with CMT or SMA, but with the same exact mutation. Even though the families had different clinical diagnoses, all the affected people had the same NEFH mutation, and it co-segregated perfectly within each family.
That's interesting, but couldn't this just be a coincidence? How is it a founder effect?
Well, there are 3 main reasons. First, all 4 families are from the same place in Japan. Second, they all have the same exact rare mutation. Third, they also all have the same haplotype around the NEFH gene, which implies that instead of this mutation randomly arising 4 separate times, it was more likely that it was inherited from a single ancestor.
Oh, you know what? It does make more sense that the mutation is a founder effect rather than multiple de novo cases. Another key assumption of the Hardy-Weinberg equilibrium is random mating. Random mating refers to the lack of preference for certain genotypes, so everyone has an equal chance of pairing. But mating isn't always random.
A key example is consanguinity. Consanguinity describes when related individuals reproduce. This skews allele frequencies by increasing rates of homozygosity. So, if a family were predisposed to a recessive trait, then consanguinity would theoretically elevate the risk of receiving both disease alleles.
Exactly. In a clinical setting, this can present as de novo variants for dominant conditions or inherited variants for recessive conditions. The latter is usually flagged for cases involving consanguinity, like that one case we learned about with the 3 affected siblings who presented primary microcephaly.
Primary microcephaly? I don't think I have that in my notes.
Essentially, primary microcephaly is a recessively inherited static neurodevelopmental disorder. They specifically found a variant in the RBBP8 gene, and right off the bat, it seems to be a frameshift mutation resulting in a premature stop codon.
Is this common in the population?
This specific variant hasn't been observed in the general population, so it could have arisen due to the complex family history.
So the condition would be sort of rare too, right?
I mean, kind of. In general populations, primary microcephaly has a relatively low incidence, but in certain populations, like those that are isolated, there is a slightly higher incidence rate.
Pause. So the concept of consanguinity elevating risk makes sense, but how exactly would we be able to measure that risk?
To do that, you'd have to go back to counting pathways on a pedigree. Trust me, it's easy once you get the hang of it.
If you say so, let's start with a simple example. Say first-degree cousins who are expecting a child. How would we count pathways in this case?
You want to start with each parent and work your way up to their shared ancestor. The biggest tip I have is that while you're traveling to the common ancestor, you can only go up. Once you reach that common ancestor, you can only go down. You should never go across the same generation.
Alright, let me give it a try. So let's start with the offspring's father. I'll pass the paternal grandfather and go up to the great-grandfather. Then I would loop back down, passing the maternal grandfather until I've reached the offspring's mother.
You got it. This counts as a single pathway. You'd repeat this but with the second common ancestor, in this case the great-grandmother. Since they don't seem to share any other common ancestors, I think we've accounted for all the possible pathways. So from here, we can calculate the coefficients of relatedness and inbreeding.
As a quick refresher, the coefficient of relatedness is the probability that 2 individuals inherit 2 same copies from common ancestors. To calculate this, you would need to count all the individuals we need to loop through until we reach the relative of interest.
And the coefficient of inbreeding is the possibility that an offspring inherits 2 of the same alleles from the common ancestors of the parents. This is half of the coefficient of relatedness. So here we pass 4 individuals in the first loop, and 4 individuals in the second loop. So the coefficient of relatedness is 1/8, which makes the coefficient of inbreeding 1/16. So here we have a slightly more complicated pedigree. I'll give you a minute to count how many pathways there are in this one.
I counted 5 pathways. So we start with the proband's father, pass their paternal grandmother, and reach their great-great-grandfather before we loop back down. We'd repeat this loop, but this time reaching the great-great-grandmother.
The next pair similarly leads to the oldest common ancestor, but this time we take a different path. We pass through the paternal grandfather, reach the great-great-grandfather before we loop back down. We'd obviously repeat this, but with the great-great-grandmother.
The last pathway is a little different. We can pass the paternal grandfather to reach a more common ancestor, the paternal and maternal great-grandmother. From here, we can loop down to proband's mother.
Great eye! A key tip when it comes to pedigree assessment is to account for all of the common ancestors. We have the obvious, the oldest common ancestors, but in special cases with more occurrences of consanguinity, we have more recent common ancestors as well. For anybody watching at home, you can try calculating the coefficients of relatedness and inbreeding here. Pause before you reveal the answers.
Wait, this is so much easier than I expected.
Loving the confidence. Let's get into something a bit more complex. In this pedigree, we have two healthy parents who we've denoted as first cousins. The proband's paternal and maternal grandparents are each first cousins as well. I'll give you a second to count how many pathways there are in this pedigree.
This one's a bit tricky. Our first pathway passes through person 1 in the third generation until it reaches person 1 in the first generation and loops back down. We have another path just like this, but passing through person 2 in the first generation.
The next pair similarly reach person 1 and 2 in the first generation. However, this time they pass through person 3 in the second generation and loop back down, passing the same individual.
The next loop passed through another set of ancestors. Instead of person 1 and 2 in the first generation, we're now looking at person 3 and 4 in the first generation. This yellow loop does this by passing through person 2 in the third generation and looping back by passing person 4 in the third generation.
And our final pair is very similar in reaching person 3 and 4 in the first generation. This time the loop is a bit different because we pass person 4 in the second generation and loop back down, passing the same individual.
For anybody watching at home, you can try calculating the coefficients of relatedness and inbreeding here. Pause here before we reveal the answers. You know what? It's all starting to click, except for the coefficients. Why do we need to keep calculating this?
The coefficient of inbreeding is useful when it comes to calculating the likelihood of an unborn child inheriting a given trait. Consanguinity goes against the Hardy-Weinberg equation, which means that we have to correct for this factor. Obviously, with different degrees of relatedness, you would get different coefficients of inbreeding, but that's a topic for another video.
For now, let's explore a case study to calculate the risk of inheritance.
Primary microcephaly has an incidence of 1 in 250,000 births, but in certain isolated populations, such as this example, the incidence is nearly 1 in 10,000 births. Given the reduced heterozygosity, what are the chances that IB1 and IB2 have a future child affected with this trait? I'll give you a moment to calculate.
Let's walk through this together. The first step is to calculate the coefficient of inbreeding, which we already did. So here we have 1 over 16. The next step is to calculate the individual frequencies of each allele. We know that the incidence of this recessive trait is 1 in 10,000. This is Q squared. From this, we can decipher that Q is 0.0001 and P is 0.999.
As I mentioned earlier, now we need to correct the Hardy-Weinberg equation for consanguinity. So we'd use the equation Q squared plus FPQ, where F is the coefficient of inbreeding. The likelihood of IB1 and IB2 having an affected child is, therefore, 0.011%.
So, do you feel ready for the midterm now?
You know what? I actually think we're gonna ace this test.